morning, students. Today we are going to discuss about factors affecting respiration. A number of internal and external factors are known to influence the aerobic respiration of plant organs or tissues. The rate of respiration, which is measured either by the rate of oxygen consumption or by CO2 evolution, is also influenced by other metabolic processes and general physiological status of the plant. The effect may be either on the exchange of gases, reflecting the effect on stomatal opening and closing, or on the metabolic process itself. Some of the internal and external factors which affect rate of respiration are described here. First is plant age and tissue types. Generally, the younger tissues have higher rate of respiration than the older ones. The mature and senescent tissue have a very low rate of respiration. This is natural because these tissues have very low anabolic activities and therefore require less energy for other cellular activities. However, when the rate of respiration of a single leaf is measured at different stages, it shows a typical sigmoidal curve. The rate of respiration is low in very young leaf and it increases in the initial stages of the development respiration development of leaf, and then it reaches a maximum when leaf is fully mature. The rate of respiration declines after that. In many mature leaves and fleshy fruits, which also show a characteristic sigmoidal curve with respect to the rate of respiration increases temporarily just before the onset of senescence or ripening of the fruit. This rise in respiration have been called climacteric. The rise in respiration in fruits has been ascribed to the increased ethylene production, which is known to induce respiration and senescence. Examples of fruits which show a climacteric rise in respiration are apple, pear, tomato, banana, etc. Some examples of fruit which do not show climacteric rise are citrus fruits, pineapple, and strawberry. The rate of respiration depends upon the tissues as well. Respiration rate of inactive tissues such as older roots and leaves, resting fruits or stem is lower than the actively growing or metabolically active tissues. Dormant seeds also have a very low rate of respiration, but the rate increase dramatically once they imbibe water and start germinating. As mentioned earlier, tissue like spadix of arum lily have a very high rate of respiration. Typical rates of respiration of some plant tissues are given below. Here we can see dry seeds, young seedling, beet slices, tomato root tip, arum, spadix, whole plant of herb. They show a typical rate of respiration. Next is availability of the substrate. As a general principle, the rate of respiration increase with an increased substrate, especially the carbohydrate availability to the respiring tissue. The etiolated leaves from dark grown seedlings have a low rate of respiration than the green actively photosynthesizing leaves. However, if the etiolate leaves are floated on a carbohydrate solution, the rate in respiration increases. Further, in a canopy, the lower shaded leaves have a low respiration rate as compared to the upper leaves exposed to the bright light. Next is temperature. Respiration, like other biochemical process, is affected by temperature. In general, within certain limits, increase in temperature result in an increase in respiration. The rate reaches a maximum value at optimum temperature and then it declines. Usually the value of Q10 between 0 and 25 degrees centigrade is between 2 and 3. That is to say, an increase in temperature within this range causes an increase in respiration by 2 to 3 folds. At about 25 degrees centigrade, here is often a decrease in Q10 and the value may reach about 1 at 30 to 35 degrees centigrade. At about 35 degrees centigrade, there is a decline in respiration is apparently because of the inactivation and destruction of enzymes involved in respiration. This may also be due to limited availability of oxygen as its solubility and diffusion decreases at higher temperature. The optimum temperature for respiration varies according to the species and also according to the prevailing environment during the plant growth. But in most tropical species, it is between 30 and 35 degrees centigrade. The xerophytes have usually higher optimum temperature than the mesophytes. The rate of respiration is inhibited at very low temperature as well. In the most of the species of the rate of respiration is almost zero at freezing temperature. However, in some plant tissue, the respiration has been recorded at as low minus 20 degrees centigrade. So आज के लिए इतना ही. थैंक यू स्टूडेंट्स.